Hi, Bio3 students. Welcome to my first attempt at screencasting. So be patient with me. I am certainly open to you guys emailing me. I'll open a discussion on Canvas if people have questions or problems. What I've got here is sort of a little video lecture to walk you through what we're going to do with the mitochondrial DNA sequences. So first thing you need to do is go to GeneticOrigins.org. In the module on Canvas, there's a whole bunch more information, and I'll point that out later. But once you get here, we are going to be focusing right now on the mitochondrial control region. So if you click on that, it will load with some history about human evolution, talking about the emergence and our out of Africa theory. I won't read that to you because I'm confident you can read that. We're going to continue to the mitochondrial control region. The theory of why we're looking at mitochondrial DNA sequences is because the mitochondrial genome is very small, just under 17,000 base pairs or individual nucleotides in the DNA. And more specifically, we're looking at this control region where there's a certain number of variations that we've been able to track over time, and even more specifically than this 1200 base pair control region through a technique called PCR, which will be a different lecture. We're looking at this 440 base pair region. So a very short region within a short piece of DNA, but because it's in this particular control region that is common to all mitochondrial DNA, variations there happen at a very specific rate. So we can use mutations in this region to predict the length of time between species. So if we continue, talks about the evolution of the genome and about what we're going to be talking about later with how we interpret our genomes to get to our proteins. And why this region? Oxygen-free radicals. We know that the mitochondria is the organelle where cellular respiration reactions like the citric acid cycle and electron transport take place. Because of that, during those electron transfers, we create radicals, and those radicals can damage DNA. So instead of having base pairing that's normal, but like a G and C, the cytosine nucleotide gets damaged, it becomes a U. That is not a correct pairing. If that pairing undergoes replication, the G might correctly pair with a C, but the U, which shouldn't be in DNA in the first place, will pair with an adenine nucleotide. And then when that replicates, we now have a continuing mutation in the genome. Continue. Now, the maternal inheritance of mitochondrial DNA, we already talked in class about how that's not entirely true anymore, but this webpage has not yet been updated. And then there's a whole bunch of information about how we can use the number of mutations in this region as a molecular clock. If the mutations happen at a consistent rate, the greater the number of mutations, the further apart those two individuals or species are in time. Now, if you look at the top, we have our GeneticOrigins mitochondrial control region. We will get to the sequence server in a different video. Right now, I want to go to Media and Animation. So click there. And we're going to go to solving the problem of the mystery of the Neanderthals. Add my coffee. So single nucleotide mutations are that molecular clock because they happen at a consistent rate. Click in the lower right to proceed to the next screen. So when we have a common ancestor, we accumulate mutations, and those numbers of mutations is proportional to the length of time we've been separate in evolutionary time. There's our common primate ancestor and its mitochondrial genome. Modern ape and us. The clock going round and round with accumulations of mutations, and we can predict how long ago those species separated. Clickety click. Now this is an image of comparison between two DNA sequences. So even though there's two lines, this is not one individual's DNA base pairing. The top line is one individual in every row, and the bottom line is a different individual in every row. So it wants us to track the number of nucleotide differences between humans and modern chimps in this part where they've compared the sequences. And we click on nucleotide differences to highlight. And this is my proof that I'm asking you to do things that I'm doing. There's a CT, those don't match. There's a GA, I already missed some. I'll go through and click wherever the top and bottom strands do not match, and they will highlight. If you click ones that do match, it won't let you highlight those. CT up. The bottom strand had a mutation, so that counts as a change or a deletion. CT. So just so you don't panic when you're going to have to be doing large sequence comparisons, that will be in a different video. The program will highlight the changes for you, so you won't have to go through 379 base pairs looking for differences. But this is good practice so that you know how that came about when it magically appears in your DNA sequence comparisons that we'll be doing later. If I'd been smart, I would have been counting as I went along, but now I'm just going to keep tracking. TC, CT. Some manner of notification just happened. Boy, isn't this exciting as we go along? CA. I'm very tempted just to count what I've got and then make a stab at the guess on the side overview, but I'm going to keep going. CT, GA. I can't believe how good my dog is being. Oh, the rain just started lashing against the window, so I'm glad I've already run my errands today. No, I just went to the grocery store and picked up exactly what I needed and got the heck out. I got AC, CT. How many do I have? 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, 27, 28, 29, 30, 31, 32, 33, 34, 35, 36, 37, 38, 39, 40, 41, 42, 43, 44, 45, 46, 47, 50. I found 51. I'm going to say 56. Sweet. So, humans and chimps have 56 average mutations. We keep clicking. Now we're going to compare this part to modern humans. They're going to be very similar. We are way more similar than we are different. I'm just not finding. Whoops, there's one. There's two. I'm counting this time. Only two so far. That seems low. They're all blurring together. Oh, there, I thought I saw a GA. Nope. 2. There's got to be more than 2. That seems too low. Up 3. It's like the world's most boring puzzle, just looking at letters that mismatch. If I say 3, 7. All right, so the number of differences has been calibrated to equal about 150,000 years. So now, 56 between us and the common chimp, and about 150,000 years between our common human ancestor. Talking about Neanderthals, where we overlapped. Now, which diagram shows Neanderthal is an ancestor of modern humans? Well, that's original ape, modern ape, so that's not different. We've got some common ancestor here, but this one shows that we have a Neanderthal split off from everything. So this one keeps the Neanderthal in the group. I'm going to say A. The guy that did the mitochondrial sequencing, and now we're going to look at the difference between a Neanderthal and a modern human. So 7 differences between 2 modern humans, and 56 between us and a chimp. I'm going to think it's going to be 19 or 28, but I don't know for a fact. Teasing. Keep going. Scroll along and find your differences. There's a deletion. AG 1, 2, 3, 4, 5, 6, 8. All right, we're already up to 8. 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 15, 18, 18. Is it 19? Then it must be 28. Wow, I did a terrible job of finding those. So don't give yourself a break if you're not finding them all. There they are. So now there are 28 differences between Neanderthals and modern humans. So if 7 mutations equal 150,000 years, how many years are equal to 28 mutations? Well, 7 times 4 is 28. So 150 times 4 is 600,000. That was an easy one. All right, so scratch my nose with my sleeve. The data are consistent with which of the 2 diagrams showing the relationship between Neanderthal and modern humans? Remember, modern humans descended from a common ancestor 150,000 years ago. And we just calculated the difference between modern humans and Neanderthal to be 600,000 years. So this one says they're further apart. And that would be a dead end. So yes, we diverged 450,000 years before the common ancestor of all modern humans. So congratulations, you're so smart. So that wraps up how the mitochondrial DNA comparison sequences were done. I'll have another video that will tell you how to actually do the exercises.